Okay, folks, good morning, good afternoon, good evening. It's me, Dr. Kaz, and we are coming down the home stretch to the digestive system. And I just wanted to kind of bring up a good point or something that you should be keeping in mind when you're studying this chapter specifically in the digestive system is when you think of the digestive system, a lot of the physiology functions of this system are based on reflexes. So keep that concept in mind. Stimulus is needed. You have to have receptors to monitor that stimulus. All right, the control center to integrate all that information, process it, and to determine what sort of outcome is going to happen. And then, of course, then finally, we need an effector, right? Something that is going to cause some sort of change, right, or an effect as a result of the stimulus. And so as we're going through this, right, a large portion, all right, of this material is based on that simple concept of a reflex here. Okay, so let's talk about that large intestine, finishing up here, otherwise known as the large bowel. All right, so we're moving from proximal to distal. And we're now approaching the more distal part of your GI tract. Right? And this structure, the large intestine, is pretty much going to be found in the abdominal cavity and the pelvic cavity. And its main job, its main responsibility it's like the cleanup batter, right? It is going to continue to absorb water, which, all right, is important because we saw a lot of water absorption occur in the small intestine, but still the chyme is still kind of a liquidy substance. So we're going to transition from that liquidy chyme more to a hardened feces. So we're going to absorb that water, compact that chyme into feces, right? And while we're at it, we're going to try to clean up some of the electrolytes there too, and anything else that we can kind of digest, that last minute digestion from the remaining material inside of your GI tract. And then eventually, right, when we're done making and compacting the feces, we're going to eliminate the feces after we've stored them for a short period of, well, I should say a short period of time, but while we've stored them in the rectum there. So the first part of our large intestine is the cecum, and it's a sac. And more importantly, it is an intraperitoneal sac. So we're going to find it within the peritoneal cavity. It's located, all right, closer to your right ileum, all right, down in the right lower quadrant there. And it is going to extend from the ileocecal valve, right? That is going to be just on the other side of the ileum of the small intestine. And so at that point, the ileum has moved some of that chyme, right, that digesting material from the distal portion of the small intestine into the cecum. Now, inferior to the cecum, we have the appendix, properly named the vermiform appendix here. Again, another intraperitoneal structure. And so it hangs off the bottom of the cecum here, right? So some of you may have had an experience with this if you've ever had to have your appendix removed, right? But it is said that we have some beneficial bacteria there that help with the digestive process there. And also in the appendix, we've lined it with lymphatic tissue. And as you know, in the lymphatic tissue, we have quite a bit of our white blood cells here. So the vermiform appendix, like I said before, is also going to be intraperitoneal, right, because it is attached inferiorly to the cecum. Now, as we move through the different portions of the large intestine, we're going to alternate, right, retroperitoneal regions and intraperitoneal regions. So we just started off with an intraperitoneal region, that would be the cecum and the vermiform appendix. Now, right, we're going to see our next section of the large intestine, which is the ascending colon. And so the colon is going to move its way superior, then it's going to traverse your abdominal cavity and then go ahead and go inferiorly. So it's going to shape an inverted U. And so again, keep in mind, these segments are going to alternate retroperitoneal with intraperitoneal regions here. All right, so the first part, the ascending colon, which is located on the right side of our abdominal cavity. It's going to head up towards the liver. That is a retroperitoneal structure. And where it's going to terminate, it's going to take a 90-degree turn, okay, and start to head towards the opposite side of your body. And that turn, right, is called the right colic flexure, also known as the hepatic flexure, because it is close to the liver. Right? The ascending colon then transitions into the transverse colon, which is going to move from the right side of your body to the left side of the body. And so it is intraperitoneal. And so the transverse colon will terminate at the left colic flexure, also known as the splenic flexure. It terminates there and it makes a 90-degree turn downwards or inferiorly. Once it does that, okay, and it heads inferior down towards your left hip, then it is called the descending colon. Again, another retroperitoneal structure, and it is going to head down, all right, towards that left ileum there, which brings us to the sigmoid colon. Now, again, it's not an exact S-shaped structure, but as the descending colon comes down, it will take a turn and then it'll head back towards the center of your pelvic cavity, right, back towards the rectum. So how I always remembered that portion of the colon that looks like the letter S, so we call that the sigmoid colon. And so the sigmoid colon, all right, is going to be found in the pelvic cavity. It is going to be intraperitoneal. And then it's going to terminate at the rectum. The rectum is basically a storage facility for the remaining undigestible material known as the feces here. All right, so the rectum is going to be retroperitoneal, and it sits there, right, in the inferiormost region of the pelvic cavity, where it is going to store the feces there. And it's loaded with smooth muscle. Because, as you know, during a bowel movement, all right, during that process of defecation, all right, that smooth muscle is going to contract and move the feces, all right, through the anus and out of your body. So here's a nice picture here of our large intestine. Okay, you have the ileum, okay, terminating here on the ascending colon, and where it connects and moves chyme into the large intestine, that is called the ileocecal valve. All right, inferior to the ileocecal valve is the cecum, and then inferior to that is the vermiform appendix. And this is all intraperitoneal. Then if we move up superiorly, all right, up towards the liver, this is the ascending colon that is retroperitoneal. Then we have our right colic flexure, or hepatic flexure, takes a 90-degree turn, and then moves from the right side of your body to the left side of your body. That's the transverse colon. That is going to be intraperitoneal. Then that takes a 90-degree turn at the left colic flexure or splenic flexure. And then it forms the descending colon, moving inferiorly down towards your left hip. And that is another retroperitoneal structure. And then as we come down to the distal portion of the large intestine, right, we kind of see this S-shaped configuration, and that is the sigmoid colon. And the sigmoid colon, all right, terminates here at the rectum. And then finally, the most distal portion of the digestive system or the gastrointestinal tract is going to be the anal canal, finally with the anus at the end there. So you'll notice, and we'll talk about some of these structures here. We've got this kind of stripe-like structure here that's called the Tinea coli. We'll talk about its function here in a moment. But you'll also notice you've got these little fat globules hanging off, right? We call those the mental appendices, and they're just literally that. They are fat tissue that hang off of the large intestine. And also you may notice that the large intestine has these little sac-like structures, and we call those haustrum. Right? One individual is a haustrum, right? The whole collective of them are called haustra. We'll talk about their purpose here in a moment. All right, then at the last part of our large intestine is the anal canal, lined with stratified squamous epithelium. Just a reminder, stratified epithelium is going to be located in areas of high mechanical stress. So you can imagine moving, all right, compacted feces from the rectum, all right, out of the anus, all right, is going to encounter mechanical stress there. So this stratified squamous epithelium is a good epithelium to have in that location because it's multi-layered. As you will see, you will get sloughed off, all right, of some of that squamous epithelium there. Now, the anal canal passes through this group of muscles that sit on the floor of the pelvic cavity. The collective name is known as the levator ani muscles. These muscles, all right, do play a role, all right, in the defecation process. Okay, we'll talk about that in a moment. Okay, but they do form the floor there of your pelvis. Inside the anal canal, you will see what we call anal columns and anal sinuses. The columns are these ridges there, and then in between the ridges are the depressions known as the anal sinuses. And of course, we're going to have lots of mucus-producing tissue there. Again, mucus, its primary job is to decrease friction, offers lubrication. So we'll see in the anal canal, we have two sphincters, all right? You know a sphincter is going to be made up of muscle tissue that surrounds an opening. You have the internal anal sphincter made up of smooth muscle, which is involuntary. And then the external anal sphincter is made up of skeletal muscle that is voluntary. So we learn how to control the external anal sphincter when we are undergoing potty training or toilet training, whatever you want to call it. So here you can see the rectum and then the anal canal here, and finally the anus, and you'll see, look at the rectum. It's loaded with muscle tissue. So that muscle tissue will contract and relax and cause the movement of the feces towards the anus there. The levator ani muscle, you'll see right here, all right, this makes up the floor of your pelvic cavity. You'll also see it extend down here, and at the distal portion of it, it is going to form the external anal sphincter. Then just internal to it, hence the name internal anal sphincter, that is made up of smooth muscle tissue, and that smooth muscle tissue, all right, is involuntary. We don't have control over that. All right, so you heard me mention, all right, the tinea coli and the amental appendices on that picture that we saw just a few moments ago. The tinea coli are smooth muscle. And this smooth muscle is going to cause, all right, the intestine to appear to have these little sac-like structures called haustra. And they're going to act like a waistband here, and we'll discuss their process here, but it helps with the movement, all right, of the material through the large intestine. The amental appendices, like I was saying before, are just these lobules, these globs of fat that hang off the outside, the external surface of the haustra there. Inside of our large intestine, we'll see plenty of different types of bacteria. And I just did a seminar that was talking about the normal flora of the bacteria that's inside of our gut. And I want to say there's 1,500 different types of bacteria. I don't know why that number is sitting in my head, all right, but I'm almost positive it's 1,500. And what they do is, all right, they help with the breaking down of our macromolecules. So we consider, all right, this type of bacteria, we call it indigenous microbiota, which stands for bacteria that, normal bacteria that's supposed to reside. We title it normal bacteria flora. This is the type of bacteria that you want to have. So when you hear people talking about, oh, I'm taking probiotics, you know, they're trying to populate, all right, their guts with this normal bacterial flora, which helps to break down your macromolecules, carbohydrates, proteins, and lipids, right? So they help in that digestive process, right? And just like any living material, they're going to need nutrients, but also they're going to process and create wastes. Well, in this case, some of the waste that they are going to produce, carbon dioxide is one of them. Hydrogen ions, again, we'll see that decreasing pH values right? And then, of course, a litany of other substances, anything from nitrogen, ammonia, right? It just varies. So this, again, helps with our process of digestion because in some cases, these bacteria are going to help produce certain B vitamins, which are essential for several of the processes that occur in your body. You've got vitamin B1, B2, B3, B5, B6, B12, all right? But also vitamin K, which is important, all right, for blood clotting. So a lot of these materials, all right, that get produced here in our large intestine, we're going to absorb back into our blood. And by doing that, then we don't have to go and scavenge, all right, for these specific materials. Now, again, I always encourage people to supplement. I used to be of the mindset that if you had a great diet, you don't need to take supplements. But folks, I hate to tell you this, just a really quick PSA, that's a public service announcement. All right, the quality of our food now compared to how it was 10, 20, 30 years ago, it's just not as good. So if you're not taking supplements, you may want to strongly consider taking supplements. All right, that being said, the end product, what comes out of the intestines is known as our feces. So in the feces, you will have some water. And in some cases, you may notice, all right, when your stools, all right, when you're having a bowel movement, you may notice that you have an increased amount of water, right? And again, that could be because there's some issue going on with the absorption processes of the water. We'll talk about some of that in some later chapters here, all right? But a lot of it, what we'll see in our feces is primarily going to be undigested material. For example, maybe fiber, some of the bacteria that has been residing in your large intestine, and epithelial cells. Well, again, think about it. As that ingested material is moving through your digestive system, right, it's going to be brushing up against the intestinal walls, and it's going to cause a sloughing off of certain cell tissue. And especially when we get down into the rectum and the anal canal, that anal canal is lined with that stratified squamous epithelial tissue. So we'll see epithelial cells in our feces. And then, of course, we'll have some salts in there. So how do we move things and mix things up in the large intestine? All right. Well, of course, when we're moving things proximal to distally, we call that peristalsis. But in the large intestine, all right, it's a little bit different. Okay. It's like a chugging machine, all right, that just needs a little bit of help. If you've ever driven an old car, you may notice it needs a little help trying to go up a hill, right? So in the large intestine, our peristalsis is going to be a little bit different than what we saw in the small intestine. It's going to be much more weak, and it'll be kind of sluggish where it just kind of chugs along. So when we're talking about movements or muscle regulation in the large intestine, right, we have Hallstrom churning and then our mass movements. Now, again, churning, we're going to be mixing material up. And so when we're dealing with the Hallstrom churning, we're going to see, all right, the Hallstrom will relax as it's filling with material. Makes sense. You don't want to contract it, right? We want it relaxed as material starts to move into the Hallstrom there. And then what we'll see as the Hallstrom starts to fill up, it's going to stimulate, all right, a reflex. Here's another reflex, like I was saying before. As it's filling up with the ingested material, with that chyme, all right, it stimulates a reflex, which causes the muscularis layer to contract. And as it does so, it churns over the material, exposing more of it, right, so we can absorb as much of that material that's in there. And then also, as that's occurring, we're going to move that material further along our digestive tract to the distal Hallstrom, right, further along, maybe in the sigmoid colon or in the transverse colon to the descending colon. Now, you may notice sometimes, right, after a meal, you have to go to the bathroom, right, or you can feel something down in your belly really start, I don't want to say the stomach rumbling, all right, but something down low, right, below the belly button, you'll feel some movement going on down there. We call those mass movements. Here's where the Taenia coli really start to get involved. So they'll elicit these strong contractions and really push, all right, that compacted fecal material into our storage facility, the rectum. Usually this is going to occur after a meal, and it can happen anywhere from 2 to 3 times a day. All right, it'll push that material through, all right, getting it into the rectum, right, and then so you can have that defecation reflex occurring there. So like I said, all right, the GI tract has a lot of reflexive movements occurring, right? We talked about several of them. Another one to add to our list is the gastrocolic reflex. So as you start to eat food, and food starts to, or I shouldn't say food, not just food, but ingested material, and your stomach starts to fill up and it starts to stretch out. This is the one that I was telling you about, that later on, all right, you may have to go to the bathroom. That's because, right, that stomach distension causes mass movements in the large intestine. And basically what we're doing is we're just making room. We're making room for food, all right, for ingested material to move along through our GI tract. And then finally, our last reflex is the defecation reflex. So again, trying to make room, we're moving things distally in our GI tract. We are going to start to move material into the rectum. And as the rectum starts to fill up, you have baroreceptors in the rectum that feel that pressure of that of the feces start to increase and increase and increase. Well, this is then going to send sensory information to the spinal cord. To the spinal cord. We don't even need to really involve the brain yet, the brain per se. We will in a moment here when we actually do the actual act of defecation, right? But what will happen is, the sensory input comes from the baroreceptors in the rectum, go to the spinal cord, right down in the sacral region of the spinal cord. It doesn't have to go too far. And then, all right, the spinal cord sends motor output via the parasympathetic nervous system to the distal portion of your large intestine, which includes the sigmoid colon and the rectum. And so what we'll see then, all right, we will see that material start to move, all right, and you get that urge to defecate, which we'll talk about here in a moment. So the actual act of defecation, going to the bathroom, and we say voluntary defecation because it's not really voluntary until you learn how to control the external anal sphincter, right? So this usually happens around the toilet training period of your life, right? Around age 3, could be sooner, could be later, okay? But this is going to involve 2 things. One is the Valsalva maneuver. The Valsalva maneuver is when you increase your abdominal pressure inside your body against what's called a closed glottis, okay? So it's just like taking a deep breath in, and if you've ever gone to the doctor's office, they'll say, take a deep breath in and bear down and push out like you're gonna go to the bathroom, but don't go to the bathroom. That's the Valsalva maneuver. You're gonna increase your abdominal pressure inside of yourself. As you are doing that, okay, when you actually want to defecate, right, then you will relax your external anal sphincter. The external anal sphincter is made up of skeletal muscle. You have voluntary control over that. I got a nice picture to show you here. All right, so this picture here is showing us, all right, what I just talked about. So here's your fecal material sitting in the rectum, okay? The baroreceptors are going to monitor the stretch on the walls there of the rectum. So as more material starts to fill up, that's going to increase the stretch. It's going to increase the nerve signals. So the nerve signals, right, go from the rectum here to the sacral portion of your spinal cord. And so the sensory neurons are going to synapse onto parasympathetic motor neurons. And those parasympathetic motor neurons now are going to send motor output information to the smooth muscle cells here, right, along the wall of the rectum. So they'll start to contract and start to move and squeeze the contents towards the anal canal. Now, as long as you have adequate, all right, voluntary control over the external anal sphincter, you should not have a bowel movement. You should not be defecating. Now we're going to recruit your cerebral cortex. We're going to bring in your frontal lobe, and we are going to recruit, all right, the brain here. So nerve signals coming down from the cerebral cortex, all right, will then stimulate somatic motor neurons, right? Somatic means you have voluntary control over that. Somatic motor neurons are going to stimulate skeletal muscle. So the somatic motor neurons then are going to cause relaxation of the external anal sphincter here. So then now the fecal material can move out of your body. At the same time, all right, and we've all done this, and you know, when you are defecating, you might have to, quote unquote, bear down, all right? And that's going to be eliciting the Valsalva maneuver. You just start to kind of push down, right? And that will increase the pressure in your abdomen, and that will also help to move the material out of your body. That's defecation. Okay, so we pretty much covered all the reflexes. I do want to just kind of talk about, all right, the individual macromolecules here, and then we'll finish up, right, with chapter 26. I know it's a long chapter, but there's a lot of stuff that goes on with digestion. All right, so let's start off with carbohydrate digestion. As you know, with carbohydrates, we have monosaccharides, disaccharides, and polysaccharides. Monosaccharides are our simple sugars, all right? And then our disaccharides are going to be some of our common sugars, like our table sugar, like sucrose, all right? Lactose, sugar that's found in milk products. All right, polysaccharides are going to be our starch, all right? And so, and another type of polysaccharide is glycogen. All right, so when you start to ingest materials, all right, for example, starch, I always think of a potato because we just had St. Patrick's Day, all right? We are going, ideally, again, undergo catabolism. We are going to start off with something big and break it down into something small. So our starch is going to be broken down into, our goal is glucose molecules. That is a monosaccharide. All right, so starch will be broken down into glucose molecules. Again, disaccharides, depending on the ingredients of our disaccharides, whether it's 2 glucose molecules, all right, or 1 glucose molecule and fructose, or 1 glucose molecule and galactose, depending on what we're dealing with, right? We're going to break down disaccharides into the individual simple sugars, the monosaccharides. So the 2 main digestion places are going to be in the mouth and in the small intestine. Now, we saw when we were talking about the oral cavity earlier on, right, that your salivary glands are going to release salivary amylase. And it's that amylase there that is going to start to break down the bonds between the glucose molecules in the starch. So problem is, well, it's not really a problem, all right, but we start to break down the starches in the mouth. Then when we swallow the starches and form that bolus as it goes into the stomach, all right, the stomach pH, because it's so low, is going to deactivate, right, that salivary amylase there. So it puts a pause basically on that digestive process there. No big deal, okay? Because, right, we're going to get that the starch or those glucose molecules, those carbohydrates, excuse me, once they get into the small intestine, we'll pick up where we left off. So we won't see any new, all right, digestion of carbohydrates in the stomach because we don't have carbohydrate enzymes produced in the stomach. So we pick it up, all right, where we left off when we get into the small intestine. Thank you, pancreatic amylase. So the pancreas is going to release, make and release its version, all right, of an enzyme to break down our carbohydrates, right? That's found in the pancreatic juice. And so we will continue on with the digestion of starch. So again, we are going to make much smaller strands of our carbohydrates. And even in some cases, we'll be able to cleave off individual pieces or monomers of glucose. So I love this picture here because it kind of basically shows you, I'm going to just zoom up here to the top here. All right. So we've got all of that stomach contents get dumped here into our small intestine, right, in the form of chyme. So we've broken down parts of the starch, and we might have, you know, some polysaccharides available, some disaccharides, and if we're lucky, maybe we can have, you know, a couple monosaccharides. But no worries, the pancreas here is going to pump in its pancreatic amylase there, and it's going to continue on, all right, with the breakdown here of our carbohydrates. So that pancreatic amylase now is going to start to make these longer chains even smaller. And now this is where we get into the brush border enzymes. If you're not familiar, if you don't remember what the brush border is, you can kind of see these speed bumps here. Those are called plicae circularis, right? They're these circular folds that are found in the stomach. But the brush border is the name that we give to microvilli on the epithelial tissue. And this whole internal lining here is loaded with our non-ciliated, right, simple columnar epithelium. And that simple columnar epithelium is going to have microvilli on the top. And that's where these brush border enzymes are going to be found. All right, so what are the brush border enzymes? All right, we've got a couple. So we're going to go with some of these names here. All right, I'll take it nice and slow. So in our small intestine, we have these partially digested starch molecules here. The pancreatic amylase then converts it into oligosaccharides, which are shorter chains than the partially digested starch. All right, we'll break it down into oligosaccharides. Like I said before, it can break it down into disaccharides or the individual glucose. Our goal is to get individual glucose or monosaccharides, ideally, okay? So dextrinase and glucoamylase, right? These guys are going to start snapping off glucose molecules off of these oligosaccharides here. So they'll just start chopping off. Here's a glucose, then we'll go after the next one, and then the next one. So they start snapping that off. Maltase. Now keep in mind, a lot of these enzymes are named after, right? Are named after the sugars, right, or the carbohydrates that they interact with. So maltase is going to break down maltose into, right, two glucose molecules. It's a disaccharide. So maltose is made up of glucose, two glucose molecules. All right, so those enzymes will help to complete that starch breakdown. And so ideally, we want to wind up with those glucose molecules. All right, well, we're not going to stop there, okay? Because don't forget, we have, all right, two other, all right, sugars or disaccharides that we want to talk about, right? We have lactose and sucrose. Well, it's nice because each of those have their own brush border enzyme. And guess what? We name those brush border enzymes, right, after, right, the disaccharide to which they work on. So, for example, lactase is going to go to work on this bond right here. So lactase is going to break that bond in the lactose molecule between glucose and galactose to provide us with these two individual monosaccharides. And then, of course, sucrase is going to break this bond here in our sucrose molecule to give us glucose and fructose. Now, you may have heard the term that if you are deficient in these enzymes, and you've heard people say, well, I'm lactose intolerant. And basically what they're saying is, I don't make enough of lactase. So I have a problem, all right, with breaking down that bond between glucose and galactose, right? Especially if I'm ingesting a lot of dairy products. So once we've broken down, all right, these starches, these oligopeptides, these disaccharides into the monosaccharides, what we're going to do is then simply just absorb those monosaccharides, all right, across the epithelial tissue there that lines the small intestine. And then we're going to just drop all those monosaccharides into our blood. So it goes into our general circulation. Now, pretty much everything is going to wind up here at the liver. Okay. Now, fructose and galactose, guess what? The liver is awesome about it. It's going to take those monosaccharides and turn them into glucose. Right? Remember, cells love glucose. Glucose. We love glucose so much, we've named chemical processes after them. Okay. Glycolysis, right? Glycogenolysis, right? Glycogenesis, all that, even gluconeogenesis. All right. We always want glucose. So fructose and galactose in the liver will be turned into glucose. Now, if we have, all right, low energy demand at that time, then those glucose monomers that we've just absorbed, we can store those in tissues. Where are we going to store them? We're going to either store them in the liver or skeletal muscle cells. But if they're all happy and they're doing great, all right, and our energy demands are low, we can also store glucose as fat, because we'll turn it over to fat, convert it over to fat, and then we can store it in adipose tissue. Now, some of our polysaccharides, you heard me mention starch, but another polysaccharide is cellulose. All right, this comes from the plant cell wall. If you've taken the general biology course, you should know the difference between an animal cell and a plant cell, okay? Well, plant cells have a cell wall. Unfortunately, we cannot break down that cell wall. We just do not possess the chemical digestive enzymes to break that down. We can break it down somewhat with mechanical digestion, actually, when you're chewing your food, okay? But you can't chemically break it down. So we refer to that as fiber. And fiber is the bulk that helps us to, quote unquote, keep us regular, helps to move, all right, material through the lumen there of your GI tract. All right, protein digestion. Let's move on to our next macromolecule, proteins. All right, proteins can be a bear, but we'll talk about how we are going to break down proteins here in a moment. Keep in mind, when we are undergoing digestion, we are starting with a big molecule, a big structure, and we're going to break it down into a smaller structure. So our proteins, all right, what are the building blocks of proteins? Amino acids. Amino acids. You should also know that amino acids are bonded together by peptide bonds. So when we are digesting our proteins, we're trying to break them down into individual amino acid units. So we need to find enzymes that can break down these peptide bonds. So we've got a couple, all right? And so we'll talk through. Here's the problem though. Remember, proteins are found everywhere. We mentioned this when we were talking about the chemical digestive process in the stomach. The cells that make the enzymes, all right, for protein digestion, they have to produce those enzymes in the inactivated state. Because if they made them and they were activated immediately inside the cell, well, heck, those digestive enzymes would digest the proteins inside the cell. And so we wouldn't get anything done. So we are going to release these protein enzymes in the inactive state. And so once they're secreted outside the cell, then we have to activate them. And we saw how we did that, for example, in the stomach. We saw, all right, that the cells there, all right, in the stomach, the chief cells produce pepsinogen. That's the inactive form. And then when it gets secreted into the lumen of the stomach, right, hydrochloric acid, HCl, will then activate pepsinogen into pepsin. That low pH from the hydrochloric acid does that, all right? The cells that produce the hydrochloric acid, parietal cells. And so what does, all right, that low pH do? And we talked about briefly in other videos how, all right, I mentioned this before, our blood pH, for example, has to be carefully regulated between 7.35 and 7.45, that range there. If we become too acidic, we start to undergo protein denaturation. And basically, what does that do? We start to break down the proteins. We start to break them down. And that's a problem. You're breaking down a protein. If you denature a protein, the protein becomes non-functional. And you know that proteins do a lot of functions. Okay, so let's talk about, all right, how we're going to break down our proteins in the small intestine. We talked briefly about it, how we did it in the stomach there. All right, that low pH, that hydrochloric acid, all right, will denature proteins and start to just break them down into smaller chunks along with pepsin. Okay, now we're going to continue that in our small intestine thanks to the pancreas. The pancreas is going to produce 3 enzymes. Now notice the names here, all right? Trypsinogen, chymotrypsinogen, and procarboxypeptidase. These are all released in the inactive form. Same thing applies to these enzymes. We produce them in the inactive form first so they don't damage the tissues that are making them. Then we're going to excrete them into the lumen where we will activate them. All right, enteropeptidase. This enzyme here, right, made in the small intestine, okay, is going to convert trypsinogen into trypsin. Now we're going to get into some fun here because you're going to see, all right, this cascade. It's almost like an activation cascade here. So that enteropeptidase is going to activate trypsinogen, all right, into trypsin, the active form. Now, what happens with trypsin, check it out. It's going to activate more trypsinogen into trypsin. So it's almost like a positive feedback sort of reaction here. And then trypsin is going to then convert or activate chymotrypsinogen into chymotrypsin. And then it's also going to cause procarboxypeptidase to be activated into carboxypeptidase. So all we had to do was activate trypsin, and then trypsin just starts running around, you know, and starts activating everything. It's so excited, it even activates itself more. Okay, so that enteropeptidase really was helpful. So what does trypsin and chymotrypsin do? They're going to, again, we're going to break those peptide bonds. But here, we're going to see them breaking down those peptide bonds in specific amino acids. So we can have a long chain of amino acids, and now we can make those long chains into smaller chains. All right, we can get these smaller strands of these peptides. Then carboxypeptidase, notice this first several letters here, carboxy. And if you forgot, our amino acids, on one end of the amino acid, all right, we're going to have our carboxylene. And then on the other end, all right, we're going to have our amino end that with our sodium, not our sodium, excuse me, our nitrogen and hydrogen group there, all right? And so this goes to the carboxylene, and it breaks that peptide bond between the actual amino acid, carboxyl end, and whatever else it's stuck onto, all right? That remaining portion of the protein there. Now, peptidase, again, it will, once we start to cleave and make these peptide chains shorter and smaller, right, where we have a dipeptide, where there's 2 amino acids strung together, dipeptidase will split that dipeptide into 2 individual amino acids. And then another enzyme, I know there's tons of enzymes here. Aminopeptidase is going to help free just amino acids off of some of these peptide chains that we'll see. So again, our main goal is to break these proteins down into their building blocks, these individual amino acids. Same scenario is going to happen here with protein digestion that we saw with carbohydrate digestion. We get the monomers, all right, which are going to be monosaccharides like glucose, fructose, all right, and glucose, all right, so we can get them to absorb across the intestinal wall. Same thing here, all right, but now we're dealing with amino acids. Now we have those individual amino acids, then they can be absorbed across, all right, the epithelial tissue there of the small intestine, all right, and then we can then get that amino acids, all right, either into circulation, or the cell can use those amino acids because maybe those cells are like, hey, you know what? I have been meaning to do some improvements inside here, and I need to replace some of these older proteins here. So those cells can actually use, all right, some of those amino acids for repair inside of themselves. So here you can see, same scenario here. We're dealing with the small intestine. Okay, so the stomach has broke down some of these proteins into smaller chains here. And so now our wonderful pancreas has been producing these three enzymes, all right, trypsinogen, chymotrypsinogen, and procarboxypeptidase. And all of that is being produced. Now, in the meantime, right, out here in the small intestine, right, in these intestinal villi structures, right, we talked about this several videos ago, how we have our epithelial cells that have, right, that microvilli here, right, and so they'll produce, right, enteropeptidase, which activates trypsin, which then trypsin will activate all three of these enzymes into their active enzyme form, and then they'll go to work on the various peptides that are found here in the small intestine. So we'll start to break down, all right, these protein chains here into peptides, smaller peptides, and then we'll involve our peptidases, and they'll come in here and they'll break down, all right, bonds between two amino acids specifically, or they'll just start cleaving off, all right, amino acids off of the peptide chains here that are in existence. Basically, what we're trying to do is break down those proteins into these individual amino acids. Then we can absorb these amino acids across here, the epithelial tissue. If the cell needs to use it for anything, they've got amino acids there, or they can then transport and absorb the amino acids into the blood, which will then get carried out throughout your circulatory system and go to certain tissues, all right, and go to the liver, all right, for plenty of different processes. Amino acids, if you wanted to make glucose through the process of gluconeogenesis. Okay, let's, the last one I want to talk about is lipids, fat digestion. And we will be talking about some of the metabolism in more detail, right? We'll talk about the good cholesterol and the bad cholesterol, not in this chapter, but when we do the metabolism chapter, it's chapter 2, 3, and 27. We'll talk about the metabolism for fats, but let's just talk about lipids. Keep in mind, all right, lipids are fat soluble. They are not water soluble, all right? So they need help. They need help, and that's basically what we're going to talk about here, right, when we're talking about lipids. So some of the different types of lipids, we've talked about triglycerides, cholesterol, steroids, eicosanols, all right, phospholipids there. So triglycerides are made up of the glycerol backbone and 3 fatty acids. Right. So we are going to have enzymes that are going to break the bonds between the glycerol backbone and the fatty acid chains there. Now, the nice thing about cholesterol is we don't need it to be broken down for absorption, depending on what your dietary intake is. And I do talk about this in another video, all right? Will determine how much cholesterol your body makes versus how much is ingested there. And you'll know what I mean when you watch that other video. So in our mouth, all right, we produce, all right, our first enzyme to break down fats, and that's lingual lipase. So it starts off in the mouth there, okay? And so when that material, those lipids reach the stomach, they will then be activated. all right, our gastric lipase produced by the chief cells here are going to start to break down, okay, those triglycerides into what we call a diglyceride. So basically, and this is going to be somewhat of a limited picture. Here's one fatty acid, here's a second fatty acid, here's a third fatty acid. That's our triglyceride molecule. This is the glycerol backbone here. So what will happen is the gastric lipase is going to cleave off one of the fatty acids. So you'll have a free fatty acid chain just hanging out by itself. And so that's what we're going to see here in our stomach. Then the stomach contents get, all right, moved into, all right, the small intestine, and then our pancreatic lipase, right, is going to be released into the duodenum there. And then we are going to continue the digestion of our triglycerides into what are known as monoglycerides, right, which will then be like this. We'll have one glycerol molecule with a fatty acid attached to it, and then boom and boom. Two fatty acids that are just off all by themselves. So one of the problems is because, again, we are dealing with lipid soluble, or excuse me, non-water soluble structures here, right? What we need to do is when we are undergoing lipid digestion, we're starting out with something big and making it into something smaller. And so when we're dealing with our large lipid droplets and we want to get them into smaller droplets, we are going to undergo this process of emulsification. This is where our bile salts come into play. They are going to help with that process of emulsification, right? So the bile salts come into play, and what they are going to do is essentially surround, all right, this fat droplet. And so bile salts are essentially going to be, you've heard this term before, amphipathic molecule, which means, all right, that molecule has a polar component to it and a non-polar component to it. And we saw that with the phospholipids, all right, with the polar head and the non-polar tail. All right, well, same thing here with our bile salts. We're going to have a polar head and a non-polar tail. So if you have your fat globule here, all right, the polar head will face out and the non-polar tail will face in. So that, and I'm not going to go around and draw the whole thing because I got a picture that will show you. So they'll just attach themselves like that. And so they'll surround that fat dropule. And when the bile salt surrounds it, all right, we form what's called a mycel. And this will allow, all right, that pancreatic lipase enzyme there to get at that molecule. So we can, again, break it down. Now, again, if you haven't seen my other video on chapter 2.3.27, all right, that talks about the metabolism here. And it gets into a little more detail about bile salts. Bile salts, your body likes to recycle those. So most of the bile salts, after the absorption process, most of your bile salts will get reabsorbed by the ileum. All right, pretty much 90% of your bile salts get reabsorbed by the ileum. The other 10% get excreted out through the feces there. But like I said, the majority are going to be recycled in the ileum or reabsorbed in the ileum so we can recycle them and use them again. Okay, so how do we undergo this whole process here for that lipid absorption? It's a little bit different than what we saw before here, right? So again, we're still going to deliver, all right, these lipids to the epithelial cells, right? But once this occurs, the bile salts, the majority of the bile salts, all right, are going to stay in the lumen. And I don't have, well, I have a picture here. I'll show it to you in a second here, okay? The bile salts will pretty much peel off of that lipid. The lipid will enter the cell, right? But the majority of those bile salts will stay, not the majority, but they'll all stay in the lumen there, where 90% get absorbed in the ileum, 10% move through the digestive tract and get excreted here. So once inside these cells, right, we're going to reform, all right, or recreate those triglycerides. So we're going to have our glycerol backbone. We're going to add on 3 fatty acids to it. Here, okay, we need to prepare our triglyceride for transport because we're going to export these triglycerides into your blood. And so to do that, we're going to wrap them up with protein. We're going to wrap them up with protein and send them out, okay? So how do we do that? We take these proteins, we add them on, okay, to these triglycerides. We'll even throw some cholesterol in there for good measure. And then we form what's called a chylomicron. And so these chylomicrons will then be released into, all right, your body back into your blood or the lymphatic system. That process of releasing it from the cell is called exocytosis. We talked about the lacteals, all right, back when we were covering the lymphatic system there, okay? So a lot of the fat there, the lipids, will be sent into the lymphatic system. It all eventually drains back into your bloodstream anyways, right? But it will go from the lymphatic system into your blood, and then eventually, once it's in the blood, it's going to wind up in the liver or some of the other tissues like adipose tissue. Let me show you the picture. Okay, so here we are. We've got our lipid droplets. They just came out of the stomach. They're fresh, ready to go. They want to hang out and do some stuff, but we've got to break down, all right, these lipid droplets, all right, into from larger lipid droplets to smaller lipid droplets, where they have to undergo that emulsification process. That's where, all right, our bile salts come into play. They will do that for us. So these bile salts, right, here is that amphipathic molecule, okay? You got your polar head, nonpolar tail. The tails face in, attach themselves onto the lipid droplet where the head is on the outside. And so it forms this micelle, this structure right here, right? So these micelles then are going to travel, all right, through, all right, the duodenum here in the small intestine, and they are going to wind up, all right, coming up to an epithelial cell, and then the bile salts here will peel off, and that lipid droplet will be absorbed into the epithelial cell itself, where we are going to rebuild from those pieces there, right? Those monoglycerides, all right, and those fatty acids that we broke down earlier, right? So remember, a monoglyceride has the glycerol backbone, but one fatty acid. All right, if it's a diglyceride, it has one glycerol backbone and 2 fatty acids attached to it. So we're going to then reassemble everything into a triglyceride. But now we're like, hey, we got to send these out. It's time to package up that material, get it out of the body. So we're going to take that triglyceride and then add some proteins to it, right, along with some cholesterol, and we're going to form what's known as a chylomicron. And then those chylomicrons will be released out of these epithelial cells through the process of exocytosis and into, all right, the lymphatic system, into these lacteals. And then the lymphatic system will drain it into the blood, or I shouldn't say drain it into, but empty it into the blood, and then it'll circulate around through the circulatory system to the liver or to other tissues there, like adipose tissue, so we can deposit those triglycerides into adipose tissue. All right, so that's it for the macromolecules, right? But it's worth mentioning here, we talked about some of the other substances that are going to be absorbed during the digestive process. Let's not forget one of the most important substances in our body, which is water. Okay, this is worth noting. Most of these substances, right, water, electrolytes, vitamins, are going to be absorbed in the small intestine. The small intestine. Right, we're still going to absorb all 3 of those in the large intestine, but a majority of that should have been absorbed by the small intestine. Okay, so this is a good statistic worth noting, okay, 8 out of 9 liters, all right, of water will be absorbed in the small intestine. Whereas the large intestine, it will convert again, all right, like I said before at the beginning of this video, it's going to convert that watery chyme into a more compacted version of the feces there, right? But majority of water, small intestine absorption, right? And it does so, again, water moves across a semi-permeable membrane, which will be epithelial tissue, right? Via osmosis. Water moves from an area of high concentration to low concentration, or it goes from an area, right? It moves to an area of greater solute concentration, where there's more glucose, where there's more sodium. It's going to want to move into that area. All right, electrolytes. All right, electrolytes are going to be some, for example, magnesium or sodium. All right, again, like I said before, the small intestine is going to absorb, all right, most of those electrolytes. All right. This again depends on what you're ingesting, what your diet is made up of. Iron, for example, here, all right, very controlled absorption. You have the hormone, all right, hepcidin. All right. This hormone actually is going to inhibit iron absorption. Right. So again, if you, depending on your diet, and it also depends on, all right, certain processes in your body. But if your iron levels are high, for example, then the hepcidin levels will be increased because we are going to try to inhibit that iron absorption, and vice versa, right? If your iron levels are low, okay, maybe you have a diet very poorly managed with how much iron you're taking in, your hepcidin levels will be decreased, right? And that will allow for more iron absorption to occur. Iron is important, all right? As you know, we need iron for hemoglobin, right? That's what actually oxygen molecules bind onto. All right, vitamin absorption, all right, ADEC, A, D, E, and K. All right, this is questionable about D, whether that's still considered a vitamin. All right, I won't get into that right now, but as of right now, okay, we still do consider it a fat-soluble vitamin. All right, but these vitamins, again, will be mainly absorbed in the small intestine, right? And this will occur, right, during our fat absorption, so hence fat soluble, right? So they'll be bound inside those micelles. Our water-soluble vitamins, B and C, all right, those don't have to work through micelles because, again, fat-soluble vitamins, right, need an assistance there, or anything that's fat soluble will need assistance with absorption, right? But our water soluble, right, will actually diffuse across the plasma membrane, or in some cases, we'll see active transport. This one is worth noting, vitamin B12. It's very important, vitamin B12. We need that for the production of erythrocytes, red blood cells, okay? So vitamin B12 absorption occurs quite a bit in the ileum. That's the distal portion of the small intestine. And we are going to require our receptor-mediated endocytosis, right? And we need the help of vitamin B12 absorption. We have to have intrinsic factor. Intrinsic factor is made in the stomach by the parietal cells. All right, folks, I hope you enjoyed this long chapter and this last video. And if you have any questions, feel free to contact me. You know how to. And enjoy the rest of the day and week.